Okay, great. Yeah, thanks everyone for joining today. It's my pleasure to introduce Dr. Jemo Park to give us talk today. And I knew Jemo a long time ago when he was doing his PhD at Stanford. At the time, I was also working at Stanford. I was at GE Healthcare and as an on-site scientist at Stanford, helping them to set up hyperpolarized C13 program. And Jemo really impressed me in many ways, even starting from his PhD training. And he's always want to know every aspect of the experiment, all the way from how to handle animals and how to mix the of the pyruvate compound and post-sequence design, running the experiment, exploring new compounds, and trying different applications and doing modeling. He was doing everything. He was extremely talented. And after he got his PhD, he stayed at Stanford as a postdoc and research associate in Dr. Dan Spellman's group. And then he joined the UT Southwest as assistant professor. And I learned recently that he got an R1 funded last year in August, doing hyperpolarized C13 imaging on TBI subjects. So this is really, really exciting. And when I know we have a lot of interest in our center in glioblastoma research as well as TBI research. So I think this will be a really good opportunity to get Jemo to show us his research in these two areas using this novel molecular imaging technology, and also share with us his experience, in the human research. This is something where we're trying to set up towards in our center. As you know, we're going to have a clinical polarizer coming in later this year. So I think this will be really, really great to get his, feedback on how he worked on this journey towards the clinical research. Great, Jemo, please.
Oh, thank you. Thank you, Ifan, for first inviting me for the talk and also the very generous introduction. And also, I'd like to thank OR for organizing the seminar. So, as Ifan mentioned, I also like to give you a brief history between Ifan and I. So, we worked together a long time ago, she said. Like, I think it's in 2009 when I started my PhD work with Professor Dan Spielman's lab. At the time, I learned a lot from Ifan. She remembers incorrectly. I was not very enthusiastic. I was not very savvy on a lot of things. I was very clumsy, but she taught me a lot, and she was a great teacher, and I'm pretty sure that she still is. And it's a truly a good, great pleasure to meet her again, and also an honor to give my independent work at UT Southwestern, in front of her and excellent researchers and experts in the Martinos Center. So today's talk includes a brief introduction of dynamic nuclear polarization, or DNP, and my very preliminary efforts for clinical translation of hyperpolarized pyruvate to human brain imaging with applications in patients with mild traumatic brain injury and glioblastoma. And at the end, I'm going to present some technical, challenges and developments that we have been working on. So as you all know, MRI is a very versatile imaging modality. They can detect, different levels of details from structural information to all the way to cellular metabolism. So I've been working more on the smaller side. So my lab has been developing methods for real-time metabolic imaging, and also investigated in vivo imaging, in vivo metabolism. So unlike conventional MRI, just for those who are doing structural imaging, MR spectroscopic imaging, or MRSI, provides spectral information in addition to spatial information. So different metabolite can be, resolved as a separate image, as is shown on the right side for choline and NAA. And compared to proton, the carbon-13 is very difficult to detect. So first, the gyromagnetic ratio is just a quarter of protons, and the natural abundance is only 1.1%. So because of the detailed metabolic information that carbon-13 spectrum can give, and also the low background signal because of the low natural abundance, carbon-13 spectroscopy can be used for investigating unique metabolic information and kinetics by infusing carbon-13 labeled substrates of interest. For example, if you're interested in glucose metabolism, because background noise is signal is almost zero, so you can infuse fully carbon-13 labeled glucose to the subject, like through IV, and then you can actually detect background-free carbon-13 metabolite products such as lactate. But even with these advantages, because of the, even with, the fully labeled substrate injection for a long time, it still suffers low MR sensitivity because of the low gyromagnetic ratio. So that's where dynamic nuclear polarization comes in. So this is MR signal equation. So MR signal is proportional to the spin density, and as well as magnetic moment, which is proportional to the gyromagnetic ratio, and also the polarization. And the polarization is also proportional to the gamma, but proportional to the magnetic field, and also inversely proportional to the temperature. So dynamic DMP can be used to amplify the low carbon-13 signals by actually transferring the high polarization of free electrons under low temperature and high magnetic field to the carbon label substrate. So as shown here in the Boltzmann distribution at 3T, under 3 Tesla, at temperature at 1 Kelvin, the electron polarization reaches almost 100%, but the carbon and proton, the polarization is still like close to 0. So so this is how it works. So the first we mix the targeted substrate with a free electron. So we are going to take advantage of high polarization of free electron at low temperature and high magnetic field, and then we mix it together, and then we put it into a microwave oven under extremely low temperature, like 1 Kelvin, and also a strong magnetic field, and then irradiate a microwave to the sample. So microwave transport the high polarization from the radical to the substrate. And because it's frozen in this condition, we need to do dissolution with hot solvent to administer the solution into live animals or humans. So once it is dissolved with hot solvent, the amplified carbon signal return to the thermal state with a T1 relaxation. This relaxation can be significantly accelerated by applying RF pulses for imaging. So this is illustrating the signal drop due to the RF excitation. So due to the multi-dimensional and time-varying and transient nature of hyperpolarized signal, data acquisition needs to be extremely rapid, and also you need to be smart in terms of utilizing the hyperpolarized signals. So pyruvate was the first substrate that was demonstrated in vivo studies with the DMP. It is ideally suited for DMP studies. It reaches high polarization level pretty quickly, and it has a fairly long longitudinal relaxation time. So T1 relaxation time is over 70 seconds, and the cellular transport could be done pretty quickly and easily through the monocarboxylate transporter or MCT. And the pyruvate and the products have like a large chemical shift dispersion from each other, so it's easy to spectrally dissolve from each other. Also, metabolically, the pyruvate has advantage compared to other substrates. For example, as shown on the right side, pyruvate is the end product of glycolysis, but at the same time, this is connected to lactate fermentation or alanine conversion, and this is also an entry substrate to mitochondria through pyruvate dehydrogenase and being a precursor of TSA cycle intermediates. So in contrast, FDG, the F19 labeled deoxyglucose, which is very commonly used for PET imaging, is mostly used for detecting glucose transporter because deoxyglucose uh, actually goes through the glucose transporter, even actually going through the hexokinase, the first step of glycolysis, but is trapped after that as a deoxyglucose 6-phosphate, but it doesn't go further to the fructose. And also, you cannot differentiate the product from the injected material. How about brain? So the feasibility of hyperpolarized carbon-13 pyruvate for imaging brain metabolism has been shown by several groups. So the first advantage or the important fact of pyruvate for brain study is, the fact that it crosses blood-brain barrier. So although BBB transport is one of the rate limiting factors for HP pyruvate metabolism, metabolic imaging, it does cross BBB fairly rapidly, and by injecting pyruvate, again we can detect the LDH and ALT. The LDH is lactate dehydrogenase, ALT is alanine aminotransferase, and PDH pyruvate dehydrogenase could be assessed. So you can think like detecting lactate, alanine, the carbon dioxide, bicarbonate as in vivo enzyme assays of LDH, PDH, and ALT. So I found these images. Actually, these images were, I believe, were acquired from IFAN in like 2012 or 2013 as an ISMA abstract using one of her pulse sequences. So as you can see here, the brain is located, here, and the pyruvate was injected, so it shows more in the blood vessels, but lactate production was primarily from the brain, so indicating that pyruvate crosses BBB, and then you can see the the LDH activity within the brain. But alanine, on the other hand, was mostly coming from produced in the peripheral muscle tissues. And bicarbonate was also produced in the brain, but more in the cortex area. So in the spectrum, you can also, this is the spectrum drawn along the brain ROI. So as expected, alanine was not detected well, but under the detection limit, the pyruvate, lactate, and bicarbonate were detected. Pyruvate hydrate is not a metabolic product, but this one is chemical that makes equilibrium with the pyruvate under a physiological pH. So to apply this technique to a human brain, we need to have at least three pieces of hardware: MR scanner, and the polarizer, and R coil. The R coil should be also tuned to carbon-13 resonance frequency. So when I moved to UT Southwestern in 2016, a clinical polarizer named SpinLab, shown on the right image, was just installed next to a clinical 3T system, which gave me a great opportunity to pursue clinical translation. The scanner was a whiteboard system, which had a compromised, a compromised gradient performance and shim coils. But this choice was natural, considering other studies like fatty liver studies that are involved with larger population, larger patient subjects, and also considering that it was in Texas, so everything is big, including people. Another piece that I acquired was this carbon-13 and proton dual frequency head coil. So this one was designed and made with consultation with Ralph Ashoyan, who has a company, MR Clinical Solutions. It has three layers. The outermost layer is a proton transmit and receive quadrature coil in black, and the red is actually carbon-13 quadrature transmit, and the 8 pieces of blue indicate 8-channel carbon-13 receive arrays. So it looks pretty nice here, and it also performs pretty well. So we had all these, but by that time when I was actually testing the coils, investigators in other sites in UCSF or Memorial Sloan Kettering and the University of Cambridge, who put initial efforts, a lot of efforts in clinical translation for brain imaging, try this technique with patients with glioblastoma. The first and second images are from glioblastoma patients and healthy volunteers on the right side. So it was very exciting. I was very excited to see these images, and these were extremely insightful. But at the same time, I was a little bit disappointed. The fact that detecting bicarbonate, which is a key product in mitochondria and most of my main interest, did not show reliably in the imaging results. So the first, still now, a lot of papers are, focused on the lactate, but I was more interested in the bicarbonate detection because it's, the key or the entry key to the mitochondria. So as an initial effort, I chose slice selective acquisition of brain slices. So it doesn't give a lot of spatial information other than the slice selection, but it gives extremely reliable, the measurement of all the products. And another advantage is, I was using a very small flip angle, like a 5-degree flip angle, so it was not perturbing the injected, hyperpolarized pyruvate signal much. So this bicarbonate peak looks really reliable, but even this one was acquired with a 5-degree, and the slice 4 was didn't have a lot of the brain mass tissue, but even so we could detect pretty good bicarbonate signal. So slice 1, so instead of doing imaging, so I initially pursued slice-wise investigation, and the slice 1 was prescribed to include the ganglia and the corpus callosum, and the second slice was prescribed to include the body of the lateral ventricles, and the third slice for the cingulate gyrus valley, and the last one is for the mostly frontal parietal cortex. So because of the the rapid acquisition, so we could actually do a kinetic analysis as well. So kinetic analysis was also performed slice wise, and it was mostly for the between the pyruvate and lactate and bicarbonate to estimate the apparent conversion rate, KPL and KPB, and the in vivo longitudinal relaxation time was also estimated T1 using a three-site exchange model. And because of the negligible relevance to the cerebral metabolism, the Allen signal was not included in the model in the kinetic analysis. In addition, I also measure the time point where the maximum of each metabolite signal was achieved. So here is shown as tau. And also the product rate of each metabolite was estimated from the mean slope of the first 3 time points. Here is R, shown as R. So the tau and R are describing like a more in the increasing regime, and the actual KPL and KPB are fitting more towards the the T1 with the T1 relaxation of all the metabolites. So it was pretty well fit for all 4 different slices. And overall, both the metabolite ratios and kinetic parameters were quite consistent throughout the slices. And without knowing a core register brain structure, it was actually difficult to make some anatomical analysis. But since the coil element had limited range of detection, so this one is showing the phantom signal detected by all 8 channels. So the solid line here is combined image, cross-sectional signal change of the combined image, and this one is showing only from the one channel. So I could do, I was thinking like maybe I could do a region weighted assessment by somewhat, spatial information from acquired from coil by coil reconstruction for with the metabolite ratio. So the taking the metabolite ratio was also to compensate B1 minus inhomogeneity. So I could actually get something. I mean, this is not super accurate, but this at least these measurements are showing somewhat spatial information about this. And also these measurements were really accurate and reliable. So we can get some lactate to total carbon ratio and the bicarbonate to the total carbon ratio. Although this one is shown for one slice and for the area under the curve, but you can actually do for KPL, KPB, and also gamma, the R, the slope, initial slope, or tau, or for anything. So this one is pretty handy to do that. And since the HP data was obtained using dual frequency coil, so this is another advantage of our setup. So other sites were heavily, they're either using the body coil just for the poor spatial resolution, the anatomical reference, or they switch coil, the carbon coil with the proton coils. But in our case, we are actually doing the dual layered, dual frequency coil for carbon and proton. So we had pretty good, registration between carbon and proton images. So we could acquire, a series of nice proton images, although it was quadrupole coil. And using that, T1 weighted imaging, we could do segmentation for the white matter, gray matter, and CSF. And then we correlated the metabolite ratio to the fractional white matter, gray matter, CSF volumes. So this is without correction, and this is more like a fractional correlation. So area under the curve for pyruvate, lactate, and pyruvate in black and lactate in red, bicarbonate in blue, were positively correlated to the volume of white matter and also gray matter, and negatively correlated to the CSF volume within each slice. And pyruvate signal normalized by total carbon um, uh, also maintained the positive correlation with the fractional white matter volume uh, and the gray matter as well, but negative correlation with fractional CSF. So this type of analysis was possible by having the high resolution proton images from the corresponding slice. So after this, we moved on to imaging approach using dynamic 2D spiral CSI, chemical shift imaging, which was originally developed by Dirk Meyer and Dan Spillman at Stanford. The spiral readout was poorly performed because of the limited gradient performance, but it has relative, better advantage over, EPI type of readout that require more, higher maximum gradient rather than slew rate. So to achieve, 1.5 centimeter in plane spatial resolution, I had to use 7 spatial interleaves as shown on the left bottom, and with a variable flip angle up to 30 degree. I didn't want to use up to 90 because I wanted to save for the next time points. And with 48 echoes of spiral as shown here. So for after each excitation, we ran same spiral readout 48 times. And then this, Fourier transform along this direction, time domain, along time domain is going to give spectral information here. And with the 48 echoes, all the metabolites of interest could be resolved spectrally. And this is how it was, reconstructed. So as shown here, this is, individual coil FID data for each, interleave, each time point, each echo, and it's pretty highly, very high dimension data. And then you do the temporal domain processing like application, zero filling, and FFT, and then do key space grading, and then also do spatial domain processing, pretty similar to the temporal domain processing. And then you combine these coils because they have a different, the phase offset. And then once you do that, you can have actually very nicely combined spectrum. And this is, phase corrected for pyruvate, peak integration. This is for lactate. This is bicarbonate, but these are actually the same spectrum. And once you do that for each time point, we get time resolved pyruvate images, and this is one of the time point, and this is the lactate, and this is bicarbonate. Although it looks okay, but it's to compensate perfusion effect of injected pyruvate, you can actually do a normalization with the total carbon. And if you do that, you can clearly see that higher pyruvate to lactate conversion and higher pyruvate to bicarbonate conversion in the white matter rather than gray matter. Okay, so we had these two different like approaches, but for patient studies, we actually used the imaging approach only. So just to give a brief overview about the glioblastoma metabolism, is highly related to the metabolic phenomena called Warburg effect. The C1 pyruvate is a very suitable candidate to access tumor metabolism, which is also known as Warburg effect. because pyruvate is located here. Um, but in the tumor, pyruvate tends to convert to lactate more rather than going into the PDH flux to the mitochondria. Um, but pyruvate is located right here, so you can actually access the LDH and PDH activities at the same time. So in the tumor, this blue area that indicates the mitochondrial like oxidative phosphorylation, this is reduced, but in the lactate fermentation from glucose is increased. So we actually did several years ago. We did animal studies with cystic glioma. We implanted here, and the tumor grew pretty quickly. And then we could see increased lactate signal from the tumor area. But at the same time, bicarbonate signal was only detected on the other healthy, normal appearing brain. And if you take the ratio, this contrast is even stronger. And we try to normalize just to see whether we can actually increase the PDH activity, recover the PDH activity by injecting dichloroacetate or DCA that inhibits PDK, pyruvate dehydrogenase kinase, which inhibits PDH. So if you inject DCA, it eventually increases PDH activity. And this is how what we could observe. So this is about 40 minutes or 45 minutes after the DCA injection. So we could see almost perfect normalization of lactate to bicarbonate ratio after DCA injection. Measuring bicarbonate is very difficult because of the small, peak and the limited SNR, but it provides very important insights in cerebral metabolism. For example, Dr. Larry Rack, a collaborator in neurology at Stanford, he had a novel hypothesis that anti-VEGF therapy actually forces increased utilization of oxidative phosphorylation in the tumor tissue, in addition to have the antivascular the growth effect. So we actually tried these drugs into the tumor-bearing rat. The arrow indicates the location of the tumor. And after 3 hours later of injecting anti-VEGF, we could see the increase of bicarbonate production here. And the lactate decreased a little bit, but if you look at the lactate to bicarbonate ratio, it was more dramatic change. And this is the group comparison. So like a baseline, 3 hours, 24 hours, 48 hours later. And in the normal appearing brain stayed almost at the same level, but the tumor area decreased dramatically and then gradually increased. It was very interesting observation to see two different groups of the rats who are actually staying at the low level, and the other was staying at high level. So after I left, they continued to investigate the effect in the to correlate the lactate to bicarbonate change to the survival rate. So the non responding the group who showed low lactate bicarbonate ratio for 48 hours actually survived all the way through, but the group who didn't respond at 48 hours, post-treatment assessment actually, died pretty quickly. So this is more like early biomarker for the survival. So previous studies were, we also did a patient study compared to other studies from other groups with the brain tumor patients. We actually wanted to scan the brain tumor metabolism, before the treatment. So other studies were acquired after the treatment or the patients with recurrent tumors. This is I think, partially due to the difficulty of recruiting them. But we had awesome, collaborator in neurosurgery. So, once they have some initial radiographic diagnosis, they call me like right away, and then we schedule for this, carbon 13 proton hybrid MR exam, and usually 5 to 6 days after the original, and which is actually a couple of days before the actual tumor biopsy or tumor resection. So if you look at this, because they were patients, we wanted to shorten the actual, the MR session within 1 hour. So we including positioning and T2 weighted flare, and the initial carbon 13 pyruvate at the imaging acquisition. We did other proton acquisition, imaging acquisition, like arterial spin labeling or proton MR spectroscopy for example, like a 2-HG detection. And we actually repeated another HP pyruvate imaging before doing the gadolinium injected T1 weighted spin echo acquisition. The reason why we maintained 30 minutes between the first and the second injection was because of this the blood analysis, which was acquired from a separate the cardiac study. So as you see here, the pyruvate is x-axis is time domain, and the y-axis is carbon 13 enrichment. So right after the injection, pyruvate carbon 13 enrichment in blood is pretty high, and then it comes back about 25 minutes after the initial injection. So that's why we maintain 30 minutes to avoid any metabolic impact from the first injection to the second injection. So this is the one patient data. So far we scanned 3 patients, and this is the first subject. I'm going to only show one subject, and this subject is 71-year-old man, and the tumor slice was prescribed for based on the proton T2 weighted flare image. And it contained, contrast enhancing mass centrally and peripherally around the non-enhancing region of edema infiltration. And the core tumor mass actually showed increased relative CVF, so indicated by ASL perfusion, except the area of necrosis. And if you actually see the total carbon map, which is sum of pyruvate, lactate, and bicarbonate, you can see increased carbon 13 signal as well, which indicates the increased perfusion. So the 2 ROIs were selected to include central enhancing component, which is ROI1, and the surrounding non-enhancing non-enhancing region in the contrast enhanced T1 image, but hyper intensity with a T2 MRI. And these ROI2 and ROI4 were just the contralateral side of a normal appearing brain. So ROI1 showed increased lactate and decreased bicarbonate as compared to the contralateral normal appearing brain. And the ROI3 showed increased lactate compared to the contralateral normal appearing brain region, but the bicarbonate production was at similar levels. So 2-HG was also not detected with the MR proton MR spectroscopy in the tumor. And eventually he went through the surgical resection of the tumor 2 days after the HP exam. And we also did the histology of the tumor, and that showed that the tumor was WHO grade 4 GBM with a wild type IDH. And also it showed a high mitotic index, MIB1, greater than 50% in the ROI1, and actually it shows in red dot. And the ROI, the yellow dot here, actually showed like a low MIB1, less than 1%, which was confirmed by GFAP histology. So although this is very preliminary data, we demonstrated the feasibility of preoperative carbon-13 MRI with hyperpolarized pyruvate in, evaluation of masses that are suspected to be malignant. And we validated the carbon-13 imaging results with the surgically resected tumor samples. And we think HP pyruvate may add metabolic axis to the radiographic diagnosis of GBM, or maybe stereotactic tumor biopsy. From all the 3 participants, we found that increased relative lactate level from the region with the hyperintense, the T2 weighted flare, and the contrast enhanced T1 weighted imaging, but decreased relative bicarbonate production was only detected from T1 enhancing region, but not in the non-enhancing region with the T2 hyperintensity. So this may imply, we probably have to investigate more, but this may imply that at least some cells in T2 hyperintense regions have transitioned to the metabolic signature of malignancy. So, and we may, neurosurgeons may need to consider for tumor resection of that regions as well in the future. And the second part is actually traumatic brain injury metabolism using the same compound. So a common feature associated with traumatic brain injury is, in addition to the primary injury, is they undergoes a lot of secondary injury processes, which are very complex, and it could be minutes to months, and maybe this is a good opportunity for the therapeutic intervention. And out of these, several metabolic alteration associated with the secondary injury of TBI, I was interested in the, measuring the, metabolic changes, mostly about the mitochondrial dysfunction. Another interesting, metabolic alteration is hyperglycolysis. So, shortly after the primary injury, the increase of glucose consumption rate without having parallel increase in mitochondrial oxidative phosphorylation, which is known as hyperglycolysis, was reported by several groups for a short period of time before undergoing hypoglycolysis. So in the beginning, right after the injury, hyperglycolysis happens, glycolysis goes up, and then followed by decreased glycolysis. And the hyperglycolysis, coupled with the mitochondrial dysfunction, has a good favor of investigation of the pyruvate metabolism, which is like I mentioned, end product of glycolysis, and branch point to the lactate fermentation and the TSA cycle. So we first performed the animal studies, rat studies, using controlled cortical impact model. This is a pretty brutal, but commonly accepted model for focal injury. So sections, this is right hemisphere was impacted by this tool, and this one was acquired 24 hours after the impact. And as you see here, the mitochondrial damage was pretty bad. So like a no bicarbonate was like a pretty decreased amount of bicarbonate was detected, but lactate production was not very increased possibly somewhere between hyperglycolysis and hypoglycolysis. And we also did histology, and the sections stained with the tunnel detected late um, cellular apoptosis within the cells that have experiencing extensive DNA fragmentation. And the cellular apoptosis could be observed only with the TBI animals, and the peaks at day 2 after the post-injury. So we also did enzyme activities over several time points, and the LDH activity was significantly higher within damaged brain sections. And also PDH activity was significantly lower in the CCI damaged region than the contralateral undamaged tissue. So other groups in University of Maryland and also UCSF reported a similar observation. So, but this Maryland group reported single time point lactate and bicarbonate measurement, and UCSF group reported longitudinal observation of lactate change. So the goal of our human studies was to evaluate the sensitivity and specificity of hyperpolarized pyruvate as imaging agents for altered cerebral glycolysis, increased lactate, and mitochondrial dysfunction in TBI patients. So to avoid any complication associated with structural damages, so we recruited the early patients with a brain injury with a normal CT and also pretty mild GCS score, Glasgow Coma Scale score, and the loss of consciousness or the loss of consciousness bigger than 10 minutes. I was particularly interested in the acute stage of TBI metabolism. So we recruited patients who were injured within 10 days. So this is, we are very lucky to recruit this subject. The subject was able to come into the scan session actually 1 day after his injury. So this guy, 35 years old man, had a focal injury to the left frontal scalp, and he had GCS score 15 and no loss of consciousness. But he was scanned 24 hours after the injury, and we could see the increased, the total carbon signal, and also we increased the relative lactate signal, and the completely decreased bicarbonate signal. We also did another subject. So this subject was scanned at 6 days after the injury. He was he fell from 6 meter from a construction site scaffolding, and he had a laceration to the left medial eyebrow, and also has some subcutaneous hematoma, not in the brain, extending laterally from the laceration. So we could see increase the HP signal there, but overall lactate was not different in the impacted hemisphere and compared to the other side, but significantly low bicarbonate production in the injured site, like here. So to summarize this HP pyruvate imaging, metabolic imaging in acute TBI metabolism, we show the clinical translation of hyperpolarized pyruvate to TBI patients. We also observed altered lactate and bicarbonate production with hyperpolarized pyruvate, and increased lactate reflects hyperglycolysis, and the decreased bicarbonate implies mitochondrial dysfunction. And this is the last part. I wanted to describe some technical challenges associated with HP imaging and some recent developments from our group. So MRSI is very attractive because it not only acquires spatial information, but also has access to the spectral information. And because of that, it's pretty straightforward in terms of image reconstruction, and also often they use the same flip angle, so the analysis actually a lot more straightforward. Although we didn't talk much about the kinetic analysis here, but it has a lot of this also a lot of these advantages, like spatial resolution is poor because you have to do sampling along the spectral domain, and the signal to noise ratio is pretty low because you're using you have to divide the RF excitation for multiple excitation for spectral acquisition. So so another popular approach is using a spectrospatial RF pulse with some imaging readout. It could be EPI or SPAR-V readout. This is, I think, the first person who proposed this was Chuck Cunningham from Sunnybrook in Canada, Toronto. It's more like exciting only one metabolite using a spectrally selective RF pulse, and then do imaging readout, and then take turn to other metabolite like lactate and pyruvate. So this is our pulse that we designed, so that excites only selective metabolite one at a time, and then do a spiral readout. And this one has advantage of improved SNR and also spatial resolution, but it is susceptible to B0 inhomogeneity and signal contamination. In case that you want to suppress one peak located here, but they happen to be within the passing band. If the let's say if you are imaging the patient with a brain tumor with a large hemorrhages, or calcification and things like that. And also it requires a long RF pulse, and also the gradient, especially for us, because of the limited gradient performance, it was actually pretty long, like 20 milliseconds. So my lab further extended this type of concept to have a multi-echo spiral imaging. So after excitation, we actually acquired multi-echoes. So bicarbonate multi-echo images, lactate multi-echo images, pyruvate multi-echo images. This one was originally developed for the cardiac imaging, but we actually applied this one for other imaging method as well, because it was very reliable because of the short readout. It's only like a 10 millisecond readout, so it's almost impossible to miss any signal, even though the T2 star was extremely short for certain metabolite at certain brain regions. So the good thing about multi-echo acquisition, you can actually do the T2 star mapping, and if you have like a B0 like off resonance map, then you can potentially do T2 mapping indirectly. And also, if you can combine these, although some T2 enhancing effect, you can improve signal to noise ratio. So this is we first tested with a red heart, so we could actually get pretty good pyruvate lactate bicarbonate images. Because of the interleaved acquisition, they were actually slightly mismatched in terms of time, but we could do dynamic acquisition, and also we could do T2 star fitting. And we also applied this for the human heart. So this is the myocardial ring, and this is lactate, this is pyruvate images, and this is actually not the acquisition time. This is like echo time, so it's 17 milliseconds, 27, 36 milliseconds. So it's pretty quickly acquired by bicarbonate, lactate, and pyruvate. We also applied this for the brain. So like a bicarbonate, lactate, pyruvate were fairly well acquired reliably, but bicarbonate still suffers low signal to noise ratio. So it is probably um, like we are trying to actually optimize the acquisition length, spiral length, and the R pulse to minimize the echo time, and also considering the T2 star decay for individual metabolite. And this is another example for the kidney imaging. So, and this is a skeletal muscle, pyruvate lactate alanine bicarbonate. And this is, as I mentioned, you can calculate this is log scale of the signal decay over echo time. So you can actually do fitting for pyruvate T2 star and lactate T2 star and bicarbonate T2 star. This is from, I think, Hart. So like one more piece, so simultaneous imaging of lactate and bicarbonate could be also, done. So I wanted to maintain, better SNR by using larger flip angle on products without perturbing too much on the pyruvate. But, I wanted to make sure that, we can differentiate the contamination versus the real signal. And the simultaneous acquisition of products, like, for example, lactate and bicarbonate, especially for the brain imaging. So spectrospatial ripples like this, designed for bicarbonate and lactate only, and the flyback EPI with the controlled spatial aliasing. So we tested with the two different phantom, bicarbonate phantom and lactate phantom. And because of the off resonance effect, they actually show up in different locations along the phase encoding direction. So the lactate phantom shows up here and the bicarbonate shows up there. So if you prescribe field of view reasonably large, you can actually differentiate lactate and bicarbonate images from the single acquisition. So this is how it was acquired. This is red liver, lactate, bicarbonate. In this case, we actually tripled the field of view, so we could actually resolve all the lactate, pyruvate, and bicarbonate from the same acquisition. So this is dynamic of lactate images. Actually, I have one more. So this is super resolution carbon-13 imaging using patch-based algorithm. So since we were acquiring a high-resolution proton MRI and the low-resolution carbon-13 imaging, so we wanted to just take a chance to use this segmented information to create some possibility map for carbon-13 image super resolution. So we actually implemented this, and then we tested with phantoms. It was with two different SNR. This one is more reasonable for bicarbonate, but this is more reasonable for pyruvate and lactate. And with a better, higher SNR, we could get better, the signal reconstruction. But in lower signal to noise ratio, it was suffering a little bit of inhomogeneity or signal contamination here. Like a red is indicating patch-based, proton patch-based, carbon-13 acquisition reconstruction, and it showed reasonable, signal accuracy when it was while it was maintaining like a sharpness and on the edges. So we wanted to make sure that it works. So we did we did actually test that if imperfection with segmentation based on the proton MRI. So this one is actually missing one segment. So one compartment is missing in proton-based segmentation, but it was actually working fairly well. So unsegmented from the proton MRI was more like still suffering like smooth transition and on the edges, but it is not worsened. But properly segmented ones were showing like a sharp edges. And also we tried with a partial carbon-13 signal loss. It happens a lot with something like a shading in the carbon-13 images. But even so, it was not over filling this missing signal within, although it was within the same compartment. So we we also tried in human brain. It was not very successful, but we made just very simple assumption like a three segmentation: white matter, gray matter, and CSF. And we tried with very first uh, healthy human brain data, and this is the results. It was successful in terms of suppressing the background noise, but within the compartment it was not very like impressive. But it could be because we were doing only 2D patch based algorithm, but with a 3D um, algorithm and with a more segmentation, for example, perfusion, it may work better. So in summary, hyperpolarized carbon-13 pyruvate is safe and can be repeatedly used for imaging brain metabolism in human. And this is also useful for monitoring metabolic changes due to pathogenesis or drug treatments longitudinally. And HP pyruvate can be translated to patients with glioblastoma and TBI. So we showed treatment-naive glioblastoma patients and also acute mild TBI patients. And also, hyperpolarized carbon-13 images can be improved both in the data acquisition and post-processing. So I'd like to thank my lab members. Like I have only 3 people like highlighted here. So we are a very small group, but we are working very hard and I guess efficiently. But we also like to, I like to thank the collaborators for the glioblastoma study in neurosurgery and neurology, and also TBI study in Christopher Madden and Marco Pino neuroradiology, and the rehab, and also the collaborator in Loma Linda University, and also UT Southwestern ARC clinical research team led by Craig Malloy and Crystal and Jeannie and all the other contributors. Also like to thank GE Healthcare and Galen Reed and Albert Chen, and for Eric for the animal models, and the R coil. And I also like to thank funding support. And thank you for staying with me. Thank you.
Great. Thank you so much, Jamal. That's an excellent presentation. I've learned a lot. And it's really wonderful to see the many very interesting research project that you're conducting. And it's very impressive. Or do we have some time for questions or?
Yes,
Okay.
absolutely. So we have our first question from Alexander Lane. Alexander, can you please unmute? Yes, you already did. Thanks.
Great. Thank you, Jay. That's really, really impressive work. I think doing this clinically in these particular populations and glioblastomas and TBI patients is really impressive. So you should be congratulated. This is really, really neat stuff. I think it's something we've all been hoping to see for quite some time now in the field. One question I just had, with the TBI cases that you had done, did you also do proton spectroscopy? And if so, how did the changes or the measures of the carbon-13 lactate metabolism and that of the pyruvate bicarbonate production rather correlate with things like the proton lactate or proton measures of creatine or NAA?
Yeah, that's actually excellent question. We haven't.
Okay.
So your question was excellent, but we haven't actually correlated with the proton lactate or choline or NAA level. I think that's going to be a very interesting project, especially if you have a matching volume to compare. For example, a lot of proton spectroscopy is done with single voxel acquisition. And if we can actually come up with a reasonable acquisition scheme and prescription, I think that's going to be awesome comparison to understand better and also interpret the HP data better.
Very interesting. Great. Thank you.
Right. Jamal, I have another question. So apparently, bicarbonate signal is small. We all know that. If you inject C1 label pyruvate, um, but it's super interesting because that's sort of your window looking into the TCA cycle, right? And so, um, how about the C2 label, the pyruvate? I think you've done that on animals in at Stanford. I wonder whether you had started to consider that for human, and and from what you understand, what do you expect to be able to pick up, the enzyme activity within the TCA cycle better than doing C1 pyruvate injection and looking at bicarbonate?
Yeah, that's a great question. So C2 pyruvate, um, can give a better information about pyruvate metabolism in the TCA cycle or in mitochondria, because the label carbon follows to acetyl-CoA, and it has also 3 branches to go. It can go to fatty acid metabolism or ketone body metabolism, or it can go to TCA cycle. And based on the animal studies by myself and also other groups in UK, they detected, lactate, C5 lactate, glutamate signal production, and they compared, and also I compared the bicarbonate production to the glutamate production. So that is more like a comparison between PDH activities and the TCA cycle activities. It is very promising, and when I was graduate student and postdoc, we developed a couple of new pulse sequence and acquisition model for C5 glutamate. And also when I first came here, I was very ambitious, and we actually I purchased a lot of C2 pyruvate for human studies. We tried the C1 pyruvate for, which is technically easier, but we had so many, the problems in, acquisition SNR, and also, so I put them on the side, and then, working more on the C1 pyruvate, because I wanted to convince myself that we are detecting reliable bicarbonate before actually pursuing C5 glutamate. But we did actually try about 3 studies, or maybe more than 3, both in brain and the heart. And we could detect C5 glutamate. I didn't include here, but that was actually from like 2 years ago. But I stopped pursuing that direction because it was not. I think the C1 pyruvate should be established first before proceeding to C2 pyruvate.
Is that because with the C2 pyruvate, the spectrum become very busy because you have a lot of different downstream metabolites produced?
Well, actually, that too. But C2 pyruvate has 2 different issues. One is chemical shift displacement artifacts, because C2 pyruvate and all the way to C2 alanine, the chemical shift difference along the carbon spectrum is like almost 200 ppm. It's really large. So if you are targeting, let's say, like 1 slice right here, but other metabolite required like here, and sometimes ours acquiring like a pyruvate. I see pyruvate no lactate because lactate was acquired. So that kind of things needs to be accommodated either in the RPulse or the acquisition side, or the prescription side. You can actually prescribe 1 here, 1 here, and then you can alternate, or you can change the during the pulse sequence, you can change the center frequency of acquisition to avoid that kind of problem.
Okay.
But that's not the major reason, but more like from the very first acquisition, the C5 glutamate didn't have enough signal-to-noise ratio. I mean, I could clearly see that, but it was not at the level for the imaging acquisition.
Okay.
Dr.
Thanks.
Park, we have 3 more questions being formed. So, Ovidio, can you please unmute?
Yes, yeah. Hi, thank you for sharing the results, and it was a really great presentation. I have 2 technical questions. One is about the multi-echo spiral sequence that you presented. So how fast, how much do you accelerate the acquisition compared to the single echo sequence, or how much do you improve the spatial resolution? Is it 6 times? So you have 6 echoes, or you have less acceleration than the number of echo. And the second question is for the line noise. So I looked at how broad are the peaks is around 1-2 ppm. I've seen in most cases. So that's about 60 hertz or 30 to 60 hertz. That is that for over the entire slice, or is it over, or is it within one voxel from when you have the multivoxel acquisition? I suppose you are quite. I mean, you have a lot of room for line noise because you have more than 10 ppm or more than 5 ppm separation among all those peaks. So 1 or 2 ppm is still good enough for you. But was just wondering is how good of a shimming you normally get, and I mean the SNR for SNR for sure it would help to have better line noise.
Yeah. So about the first about what was the
about the multi-echo sequence?
Oh, yeah.
Or the,
So
like
how right.
Like how much do you accelerate compared to the single echo or the spatial resolution? How much you improve the spatial resolution?
So about the single echo, the reason why we came up with, we wanted to measure the T2 star using the, short, readout was from the cardiac imaging, we were supposed to get reliable, bicarbonate images. But, when we were using about 30 milliseconds readout, we were looking at about, I don't know, 2 or 3 out of 8 subjects showed only good bicarbonate images, and the rest didn't show any. So like, why we are looking at like no bicarbonate? I mean, the cardiac imaging, heart should produce large amount of bicarbonate. It actually shows pretty good bicarbonate, but imaging didn't show good bicarbonate acquisition. So as an image, so we wanted to find out the T2 star. And when we actually shortened the readout time to 30 to like 20, around 20 milliseconds, the success rate went up. And then we even shortened to like right now is like less than 10 milliseconds. And then now we are seeing pretty much everybody's bicarbonate images reliably. But we are not accelerating. It's just shortened readout, but we are also acquiring 6 echoes just in case we can detect more bicarbonate images. So that's one of to answer one of your questions. And the second one was the line width. So so for the imaging, the spectrum that I showed you is not single voxel. This is more like ROI averaged. So it's multi-voxel. But the reason why it shows a large line width is likely, I mean, it has multiple contributions. One is dynamic changes, like during the short acquisition time, that targeted metabolite is keep, generated or converted to another metabolite. So, there is a time varying, aspect of the acquisition, but at the same time, the slice thickness was pretty thick. It was like 2 to 3 centimeter. So there is also spatial, B0 inhomogeneity along that, slice selection direction. And also it's a multi, voxel. So, that might be the reason. I didn't correct, the B0 inhomogeneity, in plane B0 inhomogeneity, because it was minor. But I noticed that, from the brain tumor subject, there was a significant amount of B0 offset, frequency offset.
Okay, thank you so much for sharing all of this with us.
Okay, the next question from Bruce Jenkins, please.
Oh yeah, sorry. I guess we're getting a little bit late here, so I'll try and make this real quick. A great talk, by the way. I love the combination of both the animal studies and then leading into the humans, and you've really done a lot of work along pretty much every aspect of the study. So the last part, which was where you're presenting all the modifications of pulse sequences, one of the things that struck me was if you're going to use a spatial spectral pulses, then why not just use a conventional imaging acquisition, interleaved, where you can interleave, because for each one of the, frequencies. And that way you would allow yourself to image quickly in an interleave fashion, similar, without having to use these more complicated spectroscopic readouts.
Yeah, actually, we are working on that. We are working on the spatial interleave acquisition with that. But one of the to explain why this is different from the proton is because once the number of excitations is very important in HP studies, because once you excite, it doesn't recover. So if you use a multi interleaves acquisition, then let's say 4 interleaves versus 1 interleave, like a single shot, and then you have to for a single shot you can use 90-degree excitation for any products, but for multi-shot acquisition, you should divide that full MZ magnetization for either using variable flip angle or things like that. So the signal to noise ratio is going to be reduced.
Okay, thank you. I'll let it go there just because we're running out of time. Thank you.
Thanks. So last but definitely not least, Bruce Rosen.
Yeah, at least, and time is short. So just a very quick question. Again, like others have said, it was a great talk, really enjoyed it. Your BEV data, the response, was surprising in the rapidity with which you saw the changes. You saw changes as soon as 3 hours, and we don't usually think of the action of these, drugs as occurring on those kind of timeframes. What do you think's going on?
I
don't know is a fair answer. I'm just curious if you have a model for it. It just seemed that, it's very surprising. The data seemed pretty clear that something's going on, but I never would have guessed that. So I'm curious how that's changing your view of the mechanism of action of these compounds. I wouldn't have even thought they got into the brain in those timeframes, but clearly they do.
Right. So yeah, that's a good question. Actually, I think I could have answered you, better if you asked me when I was working on that paper. But as you know, as Ifan mentioned, I'm electrical engineer, and I kind of forgot. At the time I read a lot of papers to convince myself. But Larry Rack, who actually initiated that idea, was had some hypothesis that he called it like a Warburg-Folkman hypothesis.
Or the Warburg heresy, as it's been called in some quarters. Yes.
Yeah. So
that's a different story for a different time.
Right, but long story short, it's more like the metabolic changes is gonna happen before the anti-vascular and anti-angiogenic oppression happens. So that was a major hypothesis. But I can give you a better answer after.
No, that's great, and you pointed me in the right direction. That's great. Again, fantastic talk, and thank you so much for sharing your time.
Okay, thank you very much.
Great. So thank you very much, everyone. Ethan, thank you so much for doing this hosting, and Jamo, it was a pleasure. We hope to see you in person very soon, hopefully.
Yeah, hopefully. Thank you very much.
Great.
Thank you, everyone.
Thanks, Or, for organizing it.
Sure.
Okay, bye.